Hello, I'm Helen Colhoun, AXA Professor of Medical Informatics and Epidemiology at the University of Edinburgh in Scotland. In this masterclass, I want to explore with you how the increasing availability of health data is changing how we diagnose, treat, and prevent diseases. March 2020, we all know that date. The COVID-19 pandemic had started. There was confusion and panic. It was unclear who was susceptible. There was no information to guide policy. Within a few weeks of the start of the pandemic in Scotland, using the electronic healthcare records from the entire population, linked to all laboratory testing nationwide, my team was able to show that there was enormous and highly predictable variation in susceptibility to severe COVID-19. We delineated what the high-risk medical conditions were, and we built a prediction model of severe disease that had very high accuracy. This was invaluable for informing policy, for example, who needed to be in severe isolation, or whether certain drugs should be stopped. We were able to do this and do it fast because of the extensive e-health records system in Scotland. As the AXA Professor of Medical Informatics and Epidemiology at the University of Edinburgh, I want to talk to you about how the increasing availability of large-scale electronic health records and other electronic datasets are transforming health research. In this class, you'll learn why these are exciting times for researchers and discover the challenges we need to tackle. Many of the examples I'll refer to will focus on non-communicable diseases, or NCDs for short, especially cardiovascular disease and diabetes. NCDs are the most common cause of death, responsible for 41 million deaths per year globally, and this epidemic is projected to rise to 52 million deaths per annum globally by 2030. We really need research on these diseases to accelerate, and we must leverage the full potential of digital information to radically improve health. When I was a junior doctor, we recorded everything on paper. X-rays were provided as film, prescriptions were handwritten. Now many of our clinical encounters are captured in electronic healthcare records. Images are accessed in digital format, and prescriptions are transmitted electronically to the pharmacy. Because it enables large-scale studies to be done quickly, such electronic health data has the potential to transform health research. There are exciting new sources of data pertinent to health research beyond electronic health records. For example, almost all of those with type 1 diabetes in Scotland now have some type of often implanted wearable continuous glucose monitor that gives constant readouts of blood glucose without the need for finger prick tests. I just run my phone across my wearable device and it generates the data I need. When I talk about wearable devices, I don't just mean those used by people with chronic illnesses. There are, of course, smart watches like this one I've got here, giving me my heart rate, which is quite high right now. Smart patches, smart hearables, and smart clothing that give some sort of physiologic readout, such as blood pressure, heart rate and rhythm, number of steps taken, blood oxygen level, or sleep cycle. In the United States, it's estimated that 30% of all adults wear such a device every day. Think of the amount of data that is being generated constantly, that is relevant potentially to health. At the same time, there's been a revolution in laboratory methods, especially omics technology. Omics encompasses various disciplines such as genomics, proteomics, metabolomics, and glycomics, in which we characterize and quantify pools of molecules such as many proteins simultaneously that are relevant to biology. Today, we are able to measure millions of genetic variants and thousands of proteins in a tiny, tiny sample of blood or other biofluids. So the amount of digital data we can acquire on a single individual pertaining to their current and future health trajectory is growing exponentially. A game changer has been the large-scale collection of biosamples, such as blood or urine samples. This enables omics analyses, and then the combination of those data with the electronic healthcare record and other data sources. An example from my own team is the Scottish Type 1 Diabetes Bioresource, a large collection of such data and samples from people with type 1 diabetes. We also in the UK have the UK Biobank, the ongoing Our Future Health initiative. In the United States, we have the All of Us initiative. We have the China Kadoorie Biobank and similar initiatives in many parts of the world. The foresight in investing in these health record enriched biobanks is already paying massive dividends. As well as data availability, how we access, manage, and analyse data has been completely transformed by advances in compute capacity, computing science, and statistics. What's exciting now to see are the new platforms that help us mine data better. We have pandas that facilitate data wrangling and underpinning programming languages such as Python. Then there are statistical platforms such as STAN and PyMC for Bayesian inference on data. Along with these, rapid advances in machine learning and artificial intelligence mean that our ability to make new discoveries from data has changed totally from what we had in just a few years ago. For example, we've recently applied deep learning, a type of artificial intelligence, to the millions of retinal photographs from our national diabetes eye screening programme. It has shown that this approach can detect disease and predict future disease states more efficiently than just using manual grading by a human. We've shown that this can also enable tailoring how often the person with diabetes needs to have an eye check. An important enabler in this space has been initiatives in data sharing and making analytic tools open source. All told, the ever-growing rich treasure trove of data and new tools to mine data is driving forward rapid advances in understanding the causal pathway in disease, that is to say pathogenesis, prediction of future health states, and the more precise tailoring of treatment and clinical management to the needs of the individual patient, what we call precision medicine. The advent of large-scale electronic health records has really accelerated biomedical research. This is especially true when those are linked with samples and biobanks. This approach has been a major contributor to the explosion in genetic discovery, for example. The UK Biobank, with electronic health records linked to biosamples, has resulted in more than 5,000 research papers been published in the last few years, many of which describe the genetic variants that differ in frequency between people with and without a certain disease or trait. The reason that electronic health records linked to sample banks is so important is that this approach allows us to study the relationship of genetic variation and many other biomarkers such as proteins and metabolites to many, many diseases and traits simultaneously, not just one disease at a time. Studies that would have taken years to set up and run can now be done in weeks or months on much larger numbers and with many more data points. Genetic discovery allows insight into the mechanisms by which diseases are caused, and therefore how we might intervene to prevent them through developing drugs or other preventive means. For example, using body mass index profiles and genetic data from UK Biobank, people with rare mutations in a gene for a brain receptor were shown to have had half the risk of obesity. Now a drug targeting this pathway is in development. Using the electronic health record to identify people with and without liver disease resulted in identifying a protein in the liver as a critical driver of liver disease, and this is now also the target of drug development. We can also discover that a pathway that is the target of an already developed drug for a given disease is critical for some other disease. We call this drug repurposing. This is how we come to have drugs originally licensed for psoriasis of the skin now being used for Crohn's disease, a bowel disorder. It has not all been smooth sailing, however. Although initially many gene disease associations were found, it has been realised that going from such discovery of genetic association to revealing the underlying pathway is not always straightforward. In other words, the genetic architecture underlying common disease is much more complex than originally envisaged. We have moved from a polygenic to an omnigenic model of many common diseases. Recently, this has driven us to innovative ways in how to bring together genetic variation, gene expression, and protein data more effectively to reveal the core pathways. This is now bearing fruit, and I envisage a much more rapid progress from gene to drug in the next few years. Now, many of you may have heard of precision medicine. By this, we mean tailoring of prevention and treatment in a way that takes account of the specific characteristics of individuals, their disease, or their environment. Those characteristics can often be the molecular or omic profile, that is to say, the protein biomarker or other genetic marker in the person or in the disease tissue. There have been rapid advances in this precision medicine approach to cancer treatment. Many cancer treatments, such as those for lung cancer, some leukemias, melanoma, are now chosen based on the specific mutation or biomarker in the person's tumour. Another area where precision medicine advance has been made is in the field of pharmacogenomics, which is the study of how genes affect drug response. There are now more than 500 drugs for which the drug label contains information on genomic or other omic biomarkers that are relevant to adverse drug reaction or variability in response to the drug or the need to give a different dose, and so on. Much of this understanding has come from using the electronic health record to capture variation in drug response between individuals. Progress in precision medicine for other non-communicable diseases, such as diabetes or rheumatoid arthritis, has been slower. At present, for these conditions, we still use the same line drug in everybody presenting with the condition. If it doesn't work, we move on to the next drug, and so on. Large electronic health record databases and linked biobanks are key here in allowing us to learn from the variation in the clinical trajectory of previous patients. Rather than failing to capture all of that information as we did in the previous era, we're now learning how to harness it. The vision for the future is that for many more conditions, we will be able to predict which is the best drug to use first or when to initiate a treatment for a specific individual based on the biomarkers of their disease. A key aspect of electronic healthcare records is that they are longitudinal. That is to say, they can capture the health journey of a person, potentially from birth to death. This is especially true where record systems talk to each other even when a patient moves to a new location. We can use the knowledge from such health journeys to build prediction models for future disease. For example, using electronic health record data, we recently built a predictor for heart disease and type 1 diabetes that could facilitate doctors' and patients' conversations about what age to start statin therapy and other preventive drugs. These sorts of prediction models will increasingly use much wider data sources, including that from wearables. The hope is to improve the accuracy of such models as more and more data sources become available. Genetic data in the form of genotypic risk scores are increasingly being combined with other clinical data in such prediction models. This has been hailed as a step change in prediction that will alter disease screening programs for non-communicable diseases such as cancer and heart disease. However, some caution is needed as the marginal gain in prediction with such genotypic risk scores is often only useful at the very extreme of the risk score values. Prediction and risk screening present a wider set of challenges, especially getting the balance right between using some information effectively rather than creating worried well. We need to ensure that such models are learned in sufficiently diverse populations also, and we need to evaluate how such models impact on clinical decision-making as they are used in practice. Which leads me to evaluation research and how this is enabled by large-scale electronic health records. When we introduce new treatments or devices or decision-making tools, we need to know whether they are having the impact we expect. Electronic health record databases facilitate this. For example, we were able to show that the impact of our health service providing free flash monitors or continuous glucose monitors to those with diabetes in Scotland dramatically reduced hospital admissions for hypoglycemia and metabolic crisis in diabetes. Related to this is real-world pharmacoepidemiology. As new drugs get licensed, having long-term data routinely captured and analysable allows us to evaluate the safety and effectiveness of new drugs. This is important because clinical trials are often shorter term and may not have included some types of patients that get prescribed the drug, for example, older or more frail patients. By linking our national prescribing database to the National Diabetes Electronic Health Record, we were able to show, for example, that people receiving a certain type of diabetes drug had a much increased risk of hip fracture, leading to changed prescribing advice in the elderly for that drug. I've described how advances in data sources are driving forward our understanding of disease pathogenesis and are enabling precision medicine, disease prediction, and impact evaluation. This will lead us to a world of medicine where, rather than a one-size-fits-all approach, we use more precisely tailored medicines and preventive therapies that can be targeted to those most at risk and in need. Health data are highly sensitive data. I've talked about the ability of electronic health records and other large-scale health-related data to transform health research. But there's an inherent tension between maximising the use of healthcare data and guaranteeing the privacy of citizens. What approaches are being taken to find the right balance here? Well, the model varies. In the UK, we have data safe havens or trusted research environments that have been created in which trained researchers, and in some cases the private sector under strict governance and approval conditions, can gain access to such data. Importantly, alongside this, we have active programmes of public engagement to ensure public trust and support. In May 22, the EU launched the EU Health Data Space for member states to share data both for healthcare and for health research, including industrial sector research. The EU estimates that €5.6 billion will be saved over the next few years from the better use of health data for research, innovation and policy making by the formation of this health space. This is apart from even greater savings from more efficient healthcare delivery. So we are seeing more and more of these health data sharing environments being created. Typically, in these safe environments, data are provided in what we term pseudo-anonymized state. That is to say, with very obvious personal identifiers stripped off, and with other de-identification procedures being made. Some argue, however, that even this lacks sufficient safeguards for privacy, since there could be side information easily accessible elsewhere that might still allow a person to be identified. For example, if a data user knew the ages of a person's children and their dates of birth, they might be able to re-identify that person in a dataset if it contained birth records. So alternative models and structures have been proposed, such as creating synthetic or dummy data sets. The analyst here writes their code for their study on such dummy data, then passes that into an even more restricted environment where the analysis runs on the real data without that analyst having direct access. But that too is problematic. How do we ensure that such synthetic data sets preserve all of the necessary statistical characteristics, such as the relationship between different variables that are present in the true data? This is an area that needs further development. Another challenge in the use of electronic health records for research is that they require intensive data curation, including cleaning and reformatting. Health data are not clean, perfect data at source, unlike, say, banking or internet transactions. We rely on humans to enter such health data, and there will be human error. There will be ambiguity in some terms. So, for example, what exactly do we mean by hypertension? And does everybody using that term mean the same thing? There are varying nomenclature systems for disease, and there is a lack of interoperability between those systems. So making electronic health records research usable needs highly skilled data engineers working alongside those with clinical knowledge and understanding of the provenance and meaning of clinical data. Accordingly, we need more clinicians trained in informatics, and we need more data scientists with some health data training. Finally, I would note that the regulatory framework that permits the sharing and transmission and storage and usage of these sorts of data is complex. It's often labyrinthine. In the UK and EU, we have a legal framework such as the General Data Protection Regulations, or GDPR. Likewise, we have HIPAA in the United States, PIPEDA in Canada, etc. But sharing across borders is increasingly complex, and many countries, of course, have no clear regulatory framework in place at all. Progress is being made, but the timescale is slow in comparison to our advances in the ability to mine data. So I think that the regulatory tortoise really needs to catch up with the data hare. How will this world of electronic health records research change in the future? First, we need to acknowledge that many parts of the world are currently untouched by any electronic healthcare record system at all. There needs to be much greater investment in electronic healthcare record systems. Whilst the World Health Organization has a strategy to improve digitalization of health data in many emerging economies, progress is slow. Surprisingly, investing in data systems for health is not on the radar of many large non-governmental organizations. This needs to change. Even within wealthier countries, there are large gaps. Almost half of all EU member states still use paper prescriptions. But I'm an optimist, and I think we will see greater investment and a global expansion of digital data in this next decade. I think that how data are generated and how they are made available will really change. Many patient and advocacy groups are impatient at the rate of progress. More and more people want to have their own portable healthcare record, and they want to be able to share that record with whomever they wish. Apart from wearables, they want to acquire more data on themselves. People are already taking the initiative to generate biodata on themselves. Many people are already getting their own genetic sequence from private sequencing companies. One of the things that we have learned really quickly through the COVID-19 pandemic is how feasible it is for people to learn home testing. We all know how to use these now, lateral flow tests. And we learned very quickly how to use them. So more people will be self-samplers of their own data in future. There will be a shift from healthcare systems and health maintenance organisations being the sole custodians and owners of the data, to the individual being the owner. We need to be resilient to this exciting change and make options for data sharing that maximise the use of such data for research by the global body of researchers and not just single entities. Our ability to generate, capture and analyse data will accelerate even further in this next decade. If we can solve some of the investment, ethical and regulatory challenges, the impact on health will be transformative.